All right, let's jump right in. So basically, I'm going to be talking about the motivation for developing this blended genome exome protocol, or what I'll call BGE from now on, and talk through a couple manuscripts that delve into the technical development of BGE and then how we scaled and evaluated it in our first large multi-ancestry cohort, and then finally talk a little bit about some of the projects and resources that we've been looking at. Below is a nice schematic picture that Broad Clinical Labs made that hopefully gives you a little bit of a sense of what's going on in terms of the exome portion, the low-pass genome, and then blending them together to give you this current product. So prior to the development of this, the main thing that people would purchase would be kind of a deep whole exome and then a separate genotyping array. Mostly this was on the global screening array. And, that generally assayed the things that we were most interested in, which was rare coding variation, where we think you can find the most disease relevant rare variants, given the sample sizes. And then you've got genotype array, you add imputation on this, you get a nice, pretty comprehensive overview of the common variant landscape of things. But they thought we could do better. Other people were trying to do better. And, one of the issues that we saw, particularly looking at the GSA, and, we looked at a couple of chips. This was done, Alicia Martin's group, which really investigated a lot of diversity in genomics. I kind of wanted to highlight this, and this was, it's called the global screening array. But if you look at the African population, you can see compared to, say, things like the multi-ethnic array or these more expensive chips that assay, on the order of millions of variants, raw genotypes versus, probably 500,000-600,000 variants. You see this curve is not quite the same. There's a lot of stuff at very low frequencies where in Africa, which is an older population, you should see the type of distribution here with a lot more common variants. So, this kind of gives a little bit of pause. Is the global screening array really assaying things that we would like, particularly as we scale up to a lot of these diverse cohorts that we're going to be looking at? And so this kind of presented this challenge, or at least this need for something that could overcome the limitations of the current arrays that people were buying. And so the alternatives to this sort of approach is to actually just add SNPs directly into your exome array. So you could think, you have your exome capture, it captures exons. Why not just, create more probes, capture these variants? You might get stuck in the same problem if you don't pick your probes right and your SNPs right. But you could say, there might be a cheaper alternative to these two products and just say, okay, you can pull out more non-coding SNPs by just, adding more probes to your exome. The other approach is what I'll be talking about, generating low pass. And then obviously the be all end all is kind of deep whole genome sequences where you get it all, but still the price point here is still quite a bit higher than either of these alternatives. And there is option one, I will say here, Regeneron working with Twist, which is kind of the main manufacturer of a lot of the exome capture we use, does have a diversity SNP panel. This came out around 2021. So this was in the ether around the COVID era that people were moving on from arrays and trying to leverage sequencing technologies to capture all this stuff. This is out there. I don't know too much about it, but this approach is being used to just capture common variants with additional amounts of probes in the exome. Not in the exome, but using kind of exome capture technology. And then we have the blended genome exome. I have a link here. I'll click on it. I will leave, but you will see what's out here. Broad Clinical Labs, not too long ago, made a nice splash page here, kind of giving you the overview of this stuff. The clinical blended genome exome, it starts at $150. There you go. There's your price point there. I will say the difference between what I'm going to be talking about, which is more for research purposes, is that we don't have as deep of a genome. So this has evolved over time, but now you can get one that has just as deep of an exome, as previous stuff. And we pay on the order of, I think, $100 per sample for this. But as you can see, they've created more options. You can see different applications, blah, 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 and get some stuff here. If you're interested, you can go on Bro Clinical Labs and see what is here. And I'll talk a little bit more about what's provided, but I just kind of wanted to show people that you can learn more about this directly from Bro Clinical Labs. So if I just go back here. So the first thing I'll be talking about is the technical development of this. As I was saying, this started probably right around the time that everyone went on lockdown. So it was a lot of Zoom meetings and back and forth to do this. And, on the other side of things, which I didn't have any real in-depth knowledge of on the wet lab side of things, we would kind of be going back and forth. I would be doing some of the concordance analysis that I'll show, but basically the schematic here of this blended genome is to create two libraries. One is the low pass whole genomes, PCR free, looking at a few different things, and I'll talk a little bit more about some of the details. But then you've got a separate exome capture library, and this is all before sequencing. So you're pulling this DNA out, you're creating two separate libraries, running the exome capture on the PCR plus library, coming back in, and then blending at a particular ratio to run through the sequencer. And so this is what gets you that mix. Here's some gory details. I don't know half of what's being talked about here. I think the thing to take away is that during COVID, when a lot of samples were being sequenced for testing, and when the Broad kind of pivoted to an all-out COVID diagnosis testing laboratory, there was a lot of advances made, a lot of increases in throughput. And in fact, I learned kind of secondhand that a lot of the ways, particularly to get the price point where it's at, was in some of the lessons learned from doing all this stuff. So that was really kind of encouraging, a little bit of a silver lining on an otherwise great cloud of COVID. But it's not, one of the big deals with this is the fact that we can push through quite a few samples per lane of sequencing with this product. So I'm going to talk about, I think this is really important. The exome is still the exome. Things don't change all too much with that. But the big thing here, the low pass whole genomes, is that you're using imputation to really capture all those common variants back. And you can, you think about, I'll show another schematic here, but how do you impute, just random, on average 1-2 reads across the genome at any given location. So Glimpse had come out around this time. These are from the same people that made Shape It and Impute, and have a really strong track record of this stuff. And they recently came out with some low pass stuff that looked a lot better than some of the other stuff. Kai really helped me out during this development period in running Glimpse and getting things set up for that. This is kind of the schematic that you can look at, and I think it provides a nice, easy kind of detail. You go, here's what your low pass whole genome looks like, all sorts of gaps everywhere, right? And what we're trying to do is we're looking at, say, in the reference panel, there's SNPs all across here. Sometimes there's 2 reads, sometimes there's 1 read, sometimes there's 0 reads. If there's 0 reads, your prior is probably, pretty equal. It could be a het, could be a home alt, but if you have a couple reads, you can adjust those priors, and then you take those genotype prior likelihoods that you get across, say, whatever number of millions of imputed variants that you're wanting to impute, and then you kind of run through, what is kind of seen as the more traditional kind of imputation pipeline, where you phase, select haplotypes, best guess haplotypes. You run a hidden Markov model through and sample through haplotypes doing this. And then once you get these, you get your posteriors that are informed both by the phasing and imputation portion of things, of what reference panel haplotypes you think you most closely match, but also, informed by those priors based on that low pass. There's a lot more to this than just this, but hopefully that gives you a kind of a schematic of taking low pass and doing that same haplotype selection with this and then running imputation. So in these early iterations, what we used was just some deep whole genomes from a quite small sample sets that we pretty much ran through the entire tubes across these iterations. So started with 62 samples, went to 31, ran out of all their blood, took 23 samples from a different cohort. And basically what I was comparing is, taking these read files, phasing and imputation and glimpse, comparing to what we would see in GSA, running concordance with these 30x whole genomes as our quote unquote truth data set, and making sure that, seeing, pretty strong concordance with what we see there. This is kind of the final figure of this. I would say this is probably the most important figure. And these were different iterations that we did as we started with different blending operations. What you would see early on as we ran through, we kind of went way too high on the exomes. We did pretty well. The goal was to just not drop our exome coverage too much. But with the low pass genomes, we wanted to kind of hit this sweet spot where, how much coverage do we need to do pretty well to get strong concordance with our 30x whole genomes. And also on the very left here, these are just the same samples. If we pull out just the GSA SNPs, we're generally doing better across the board than we would if we imputed using just the GSA array SNPs here. But we kind of landed in this area. And so we're pretty keen, it was about 33% whole exome sequencing and 67% whole genome sequencing is kind of where we landed at as our thing. And this is where we kind of brought it into the next stage. And so I'm just going to go back to what is BGE. You've got your fragmented DNA. You've got, I just said your 1/3 exome, 1/3, 2/3 whole genome. You're going to have a lot more reads in the exome, but it's a much smaller portion of the genome. Generally, what I've seen kind of thrown around, you get a, roughly 30x whole exome, 4x whole genome. That's not quite right. It's something more spread out. Obviously, there's more variance to this. You get somewhere around 25 to 40x whole exome. This is the research kind of end of things, not that clinical deeper exome. And then something on the order of 1 to 4x whole genome here. And one of the things that I wanted to outline is that, if you plotted, this was an early pilot sample after the development, this whole genome coverage and the exome coverage here, there's some winners and some losers. Obviously, these samples would have been thrown out. This was the pilot data, but there's going to be samples in our protocol that get around 1x, and they also get the lowest amount of exome coverage. And this is just how it kind of works in the wash of high throughput. But what we really want is to just make sure that we get enough exome coverage, that we're capturing enough reads at 10x, at least 10x coverage, and then we kind of get whatever else comes out. And as long as we have enough DNA in there, we don't want to get down to 0, but these low pass imputation things, they'll work on, 0.1x, 1x. They will do better as you go up, but they will do, it will asymptote in terms of imputation performance. I won't say it's not linear, but I think once you get at least 1x coverage, you're doing pretty well in the imputation landscape. This is another thing just to show the distribution of these coverages. You get anywhere from 1 to 5x. This is a much larger cohort, and then you get around that 30x mean coding depth. And so really the goals, the deliverables, at least from the genotyping side of things, or at least the sequencing side of things, is 90% of the exome at 10x. And, we wanted to see that at least 45% of reads are going into the genome. This is a kind of another way to look at it. It's a little fuzzy, but As you can see, when you get high coverage, a 5x coverage, a much smaller proportion go to the exome, but you're getting, as long as we're getting 90% of our target captured at 10x, that's kind of the main delivery. We're giving you that deep enough exome that we're asking for. You're getting a low pass whole genome as well. Some samples do better than others, and this is something you may want to be looking at downstream to see just how much something like your low pass coverage is doing relative to, say, the imputation performance, and something to look at. But that's kind of something that might be not intuitive about this product, but this is how it works. So after the technical development, we scaled and evaluated this in a large multi-ancestry cohort, also looking at structural variants as well, which I'll show. This was kind of our first look at a collection we've been gathering for quite a while at the Stanley Center, and this is the Populations Underrepresented in Mental Illness Association, or PUMAs. And as you can see here from the names, most of the samples that we were looking at here are from the NeuroGap, and you can see various locations across Africa as well as South America. The GPC in the US, this is actually pretty enriched for African American ancestry as well. So, trying to test this across a wide diversity samples. If you look at the PC plot, it's different than what you normally see with your canonical triangle of these different things. And, as you probably look at lower lower PC loadings, you'll probably see separation of stuff, but just showing this to show it's not not your usual plot that people like to that also had deep whole genomes. And this was run using the GATK GCNV. So this is still focusing primarily on that exome content. I will say that, this doesn't in any way really turn around the low pass whole genomes as much as, well, are we capturing rare coding structural variants well enough in this new product? And you can see with the recall, so basically, how much do we see in terms of what's available, what's seen in the deep whole genomes as you get up to, say, about 5 exons overlapping, you're getting above that kind of 0.8 threshold and beyond, and you can see the distribution. So you will say most of what we see is below that. So you could say there is probably room to improve on this front. Maybe if your primary thing is to look at SVs and you're not happy with this is knowledge that you may want to have. On the other side of things, you're still, what you do collect is pretty well captured in terms of not calling a whole bunch of false positives. So what you do collect is pretty well identified in terms of this positive predictive value. But that's what we see in these samples. As you pull out to, say, that larger 50,000 sample set, we don't have this concordance that you can look at, but we just wanted to look at, say, overall distributions of deletions and duplications in these coding regions. And we compared that against Nomad v4, and largely things looked pretty good. Nothing too crazy. You do see, if anything, we're calling a little bit more here, but the main thing we worry about is that everything would be on the other side of things. If, say, you looked at the proportion of samples that do carry a rare coding deletion or duplication, we're right around the same area. A little bit lower, but I wouldn't look too much into the exact numbers here. We're roughly in the same range with what we've seen before. So there's no real like red flags that, doing this and using BGE over the exome capture that was used before is going to really hurt you in a meaningful way. You do have a little bit less sensitivity in those very small regions. So when we looked at the imputation evaluation, so what differs here in terms of what we did before when we had deep whole genomes, we have quite a few samples, but we didn't have 30x whole genomes for a lot of this stuff. So what we did instead was look at GSA array genotypes. And so kind of subsetting that fraction of the imputed calls to what we see in these same samples with their arrays. There's quite a bit of kind of back and forth QC to kind of get things right there. But we looked at, probably on the order of 11, 12,000 individuals among that sample set to look at this concordance. And I also threw this here to give a little bit of a sense of what the cost per sample is in doing this. So on average, it's about 36 cents. That's still roughly the same. There was a bit of work on optimizing how large these batches had to be and what sort of compute you're asking for to kind of get these numbers as low as we can. And that's a whole nother thing that I won't talk too much about. But during the time that we scaled up, there was also an advance in the Glimpse software. And probably the main thing is you get large reference panels. Things can get expensive with low pass whole genomes. You could say 242 pounds using, say, this other option here. The main thing, you've got $1 in Glimpse 1, 8 cents. This sounds very attractive. It's a lot cheaper to do this stuff, particularly when you're using, say, 150,000 samples as your reference panel instead of just, say, a few thousand genomes, something like that. Kind of the big takeaway from this: sparsity, 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 sparsity. Lots of rare variants in your reference panel now. We need to handle those differently. We got to leverage tools and adapt to the sparsity of this stuff. And as you can see, I think the main thing with this UK Biobank is that Glimpse 2 is doing better at those rare variants than Glimpse 1 as you get to those lower frequencies and up there with that super expensive version of low-pass imputation. Also, I won't go too much into this for time's sake, but having a large reference panel does really well across a pretty wide array. You have to get pretty far from that little island over there in the UK before you really start seeing things like 1,000 genomes and the diversity stuff starting to perform better than, say, a much larger reference panel cohort. So something interesting to think about in terms of what you choose, that probably a large reference panel can go a long way in that sense. So we did our implementation in Glimpse 2 with this stuff. We used a version of the 1,000 genomes HTTP reference panel. We did remove a lot of the rarest variants. That actually drops down. There's on the order of like 100 and some odd million, 150 million SNPs. But if you drop out those singletons and doubletons in these 4K samples, you can get down to somewhere around 67 million. We don't really trust the rarest variants in this to impute all that well. Anyways, do some QC. We look at concordance with those arrays. We see results that are largely consistent with what we were seeing in our pilot samples, which is very encouraging that, across many different ancestries and different, different groups that, we're seeing largely, we're getting above, even at the lowest frequency that we could kind of examine using this GSA array. It's not quite as sensitive as those whole genomes, obviously, but, we are still above 90% in terms of our R-squared metric. So things looked good there. One of the things I found interesting, this is in the supplement, but if you split out by coding and non-coding SNPs, that we were able to do that, you do see things. I mean, there is one little outlier here, but generally things are better. this is kind of what I wanted to see. I was never quite sure about these priors and how well that they do, but obviously this is being tagged by a lot of this deeper exome content, right? And so you're going to see that when you compare it against, these lower frequencies around. So, generally you're going to want to use the exome, but you are getting some interesting kind of, in terms of imputation, coding regions, you've got 30x mean sequencing depth. It is translating into better imputation performance. So, you're not throwing out the exome when you run through low pass imputation. You take whatever you can get. And on the other side of things, we do see higher concordance, which I found kind of interesting. We did as well look at how well you could capture structural variants in the low pass genomes themselves. I wouldn't say this was a particular use case that people thought think to do in this stuff, but it was worth investigating as well. And what you see is that as you get to a certain length, the structural variants you call do actually pretty well, in particular for deletions. Anything even in this 0 to 500 base pair range, you're doing pretty. The deletions you're calling are deletions that we're also seeing in deep whole genomes. The bad side of this is that your recall is way low, so you're not capturing all of it. You're capturing on the order of, say, 30 to 40%. And as you go to that lowest one, you're capturing a lot less. I mean, maybe if you play around with the tuning of these things, you could change things up. But I think as currently stands with the analysis that was run, it's interesting to think that there is room to capture some rare structural variant outside of the coding region, and say with some confidence that even though you're only capturing a fraction of this stuff, it's most likely real. It's not just filled up with a whole bunch of false positive calls because of the low pass. So that's a little bit of a interesting side note in these analyses. And so in the last few minutes here, I can talk about some of the current BGE products, resources. We've given talks, at least the lab has rolled a number of these BGE samples into the larger BIPEX and schema, so bipolar and schizophrenia exome meta-analyses. I will highlight, bipolar in particular, prior to the incorporation of some of these new samples, had very few, if any, exome-wide significant hits. And we're really seeing some nice results out of that. Neurogap GWAS, it's still early days with this stuff. African populations have a lot more diversity. I think we're going to be waiting on more samples to find stuff there, but it's really encouraging to see, downstream results popping out. We have a lot more samples in tow. In fact, we have probably within our group and adjacent groups, something on the order of over 260,000 samples across the globe, making this up probably the largest. That PUMAS cohort that we looked at about 50,000. It's going to be growing to about 140,000, primarily schizophrenia, bipolar, psychosis. But we have a number of other samples as well, and some different diseases that we're interested in. I know other people out there are also starting to look at BGE sample collections. In terms of running something like Glimpse, we ran a lot of this through our Hail batch system, and it's a bit more internal, but there is a version of running Glimpse 2 right through Terra. I will say if your sample size is somewhere probably around 1,000, maybe up to a few thousand, this is actually probably the best route to run things through. Once you start getting into the 10,000, there are limitations to running this. It's not that it won't work, it's that it will take weeks to run through. There's quotas on the other deep in the belly of this. I don't know why there are quotas, but there are quotas, and that's just how it is. And we get around it by running basically the same thing that was developed in GP through the R Hale batch compute. But this is out there. You know, if you click on this, you can basically see this. And one of the nice things that Michael did was basically put all the settings you just kind of fill in, rename this demo into whatever you're doing, and you know, select how much you want. The main thing, the billing project that's gonna actually pay for all this stuff, and you can do this. So I think, you know, we're really close to something that's, you know, pretty accessible to people who don't have kind of deep knowledge of kind of the inner workings of GLIMPs. We are going to be getting beyond 1,000 genomes HGDP. A lot of the samples that we've done so far, that's been the default, but there is a much larger imputation service. We're hoping that it'll support GLIMPs later this year. I think right now they're in kind of the pilot phase. We jump to this. So this combines all of us and the Anvil Centers for Common Disease Genomics. So it's about, I think, on the order of 800,000 samples. So it's big. It's got lots of variants. As many of us are coming to learn, you know, lots of variants are good until you have to store them and let them sit around for a while. But oh, here we go. We've got 515,000 that came out here. And so I think they're in the pilot phase right now. So if anybody's got arrays that they want imputed using the service, this might be a good resource to jump out to. Not ready for GLIMPs yet, but, you know, we will be kind of making a big move with a lot of our samples into this space because we really want to leverage, you know, the sample size and diversity that all of us and Anvil will give us. And also, we have a BGE toolkit. It's currently in the works. But in terms of, say, running imputation through the Hale batch version and doing things like QC and potentially, say, if you're interested, I mean, one of the things I didn't really talk too much about, you know, things still work in the, you know, impute world of things. You get your imputation, you run your GWAS meta-analysis, and then the exome people run their exome stuff. But, you know, we really want to merge all this stuff at the end of the day, having both common variants and rare variants together. Why not? We are working on merging this in ways that I think would be sensible and giving analysts the ability to kind of select just how they want to do this, and hopefully giving these sorts of tools will help here. And we are developing this now as we move into kind of the next phase of this. And I think with that, I just want to thank a lot of those within our group that helped all the way from the early days to a lot of the scaling up and recent analysis out here. It's been really an effort that spans well beyond our units, you know, with the help of those in GP. And, you know, we're hoping that as we get here, you know, this idea of diversity in genomic association studies can, you know, be realized. And I think the low pass approach really, in the long term, you know, aside from, say, the inevitable, everyone has deep whole genomes, if that cost, you know, ever truly gets down to something as competitive with this, but that, you know, you can go to basically any population in the world, and, you know, with the right reference panel, is that you're not, you know, systematically ignoring the sort of variation that may exist there, where, you know, with the array approach, you have the possibility that, you know, we are kind of underserving what we can see in the landscape of variation. And so really excited about this, and thanks.
All right, with that, let's thank Daniel one last time.